We've talked a bunch about signaling molecules such as neurotransmitters, and we've talked a little bit about hormones, specifically insulin. Our main signaling molecules in the body are hormones and neurotransmitters. Certainly they are not the exhaustive list of all of them. We have things known as paracrines, autocrines, factors, but the main ones we're going to be talking about are hormones and neurotransmitters. Neurotransmitters are released from nerve cells, known as neurons, and hormones are released from glandular tissue or endocrine tissue, which is composed of epithelial tissue. And we'll get into more detail on the endocrine system as we move forward. Any signaling molecule needs to have some sort of docking station or receptor to bind to elicit some sort of downstream response, or else there's no point in releasing those neurotransmitters. For example, we talked about the release of acetylcholine binding to the heart muscle, the myocardium, to decrease heart rate. So it's binding to a specific receptor. So receptors have specificity, meaning only certain signaling molecules, maybe just one, can bind to them. Maybe it's a few different signaling molecules. So let's take a look at this. I would say the most prevalent receptor we've talked about are ligand-gated ion channels, which we see right here. And I'm calling it a receptor, which it is, but it's both a receptor and a channel. We're going to talk about proteins on cell membranes that function only as a receptor, but in this case, this is a ligand-gated ion channel, meaning a ligand is going to bind to it. So we see a binding spot right there on this protein, and there is a gate that is closed. So I should back up and say probably all receptors in the body are composed of proteins. And these proteins are embedded within the plasma membrane of cells. And they will span the entirety of the plasma membrane. And the whole purpose, really, for a lot of these receptors are polar molecules to bind to them. Because polar molecules cannot cross the plasma membrane without a channel. And really, all these signaling molecules are trying to do is elicit some sort of activity within the cell. So what happens is a polar molecule will bind to a receptor on the extracellular fluid side of the plasma membrane and elicit some sort of response or activity. So in the case of a ligand-gated ion channel, we can see in this cell right here, it is polarized. I don't have the positives on the outside. I'm just showing the negative on the interior of the cell for simplicity. But this gate is closed. There is no ligand bound to it. This cell, or at least the receptor and channel, is inactive. If we have the signaling molecule binding to this channel, it is opening the channel, and in this case, we would call this a ligand-gated sodium channel. Sodium moves through the channel, depolarizing the cell. Now, depolarizing the cell is really an intermediate step. For example, if we think about neurons, the whole purpose of depolarizing the neuron is to release neurotransmitters. The whole purpose of depolarizing a pancreatic beta cell is to release insulin. So the depolarization event is not the end-all be-all of cellular activity. It's really just an intermediate step. But what initiates this process is a signaling molecule, otherwise known as a ligand, binding to the receptor. And the receptor is just this little circular indentation. And these receptors, once again, are very specific to these signaling molecules. It could be based on the specificity of the shape of the signaling molecules or something about the biochemistry of the signaling molecule that has a greater affinity for the protein or a diminished affinity for the protein. One thing to keep in mind is this gate is open on this ligand-gated ion channel, and it opened due to a conformational change. Once again, these receptor channels are proteins, and the binding of the ligand induces a conformational change. That is an intended change in shape of the protein, and what that does in this case, it opens a gate to facilitate the movement of something through the channel. And that something in this case is sodium, and that's depolarizing the cell. A prime example of a ligand-gated ion channel are nicotinic receptors. In a previous video, I will link that video up here, that we talked about neurotransmitters and different types of neurotransmitters, and specifically acetylcholine. Acetylcholine is going to bind to two types of receptors, nicotinic receptors and muscarinic receptors. Nicotinic receptors, or more specifically, cholinergic nicotinic receptors, are ligand-gated ion channels. The ligand is acetylcholine, and the ion, it could be variable, but in this case, it is sodium. The other main types of receptors are what we refer to as G-protein linked receptors, and admittedly, these are much more complicated and harder to kind of grasp. So I'll try to take this in small steps, and to be very clear, what I am going to display here is dramatically simplified. But these are G-protein linked receptors. So what we have in blue is the receptor. What we have in red is the G-protein. And binding to G-protein in this case is guanosine it's guanosine diphosphate that does get converted to guanosine triphosphate, GTP. GDP and GTP, which are very, 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 very similar to ADP and ATP. GTP is a very energy-rich molecule, just like ATP. So to be clear, the red thing is the G protein. The G stands for guanosine or guanine. That's the G protein. It is physically attached to or linked to the receptor that we see in blue. So it is a G protein linked receptor, often referred to as a G protein coupled receptor. And up here, we see a signaling molecule. But I'm going to tell you right now, that signaling molecule, that beautiful symmetric circle, is not going to fit in this rectangular shaped receptor. So that's a good example of specificity. Another thing I want to show in this cell right here is an enzyme. G protein linked receptors and processes involve a whole cascade of events, or what's known as signal transduction, which activates one enzyme that activates a protein that activates another protein, and it's a whole multitude of steps that we are not going to go into, but it's activated by a lipophobic molecule, as are these ligand-gated ion channels. These are lipophobic, and because they're lipophobic, well, they are lipophobic, I should say, because they are polar molecules. Polar molecules are lipophobic that want nothing to do with the plasma membrane. They don't want to pass through the lipid tail region of the interior of this plasma membrane. So they need to utilize a receptor on the outside of the plasma membrane. A little side note, nonpolar molecules, which are lipophilic, they love being around lipids, can easily cross plasma membranes without a channel or without a receptor on the extracellular surface. The receptors for lipid-based molecules and nonpolar molecules are within cells, within the cytoplasm, or oftentimes even within the nucleus of cells. But back to this G protein linked receptor. This is the receptor, that's the G protein, and that is an enzyme. When the proper signaling molecule binds to that receptor, it is going to activate the G protein. We see that GDP was phosphorylated into GTP, and because that's a high energy molecule, it activates this protein, which then further turns on this enzyme, which is going to engage in some further activity. I want to be very clear, what I've just shown here is dramatically simplified, but it's enough for our purposes for physiology. That is to say, this is a G protein linked receptor. The G protein gets activated when the appropriate signaling molecule or ligand binds to the receptor. Once the G protein is turning on the enzyme, there's a number of different options. One, it can engage in a whole cascade of events right here. And I'm not saying that this particular cell can do both options, but G protein linked processes and cascades are quite varied throughout the body. So here, I am just showing the enzyme with the G protein bound to it is going to produce cyclic AMP, which is another nucleotide, just like GDP or ADP. This is cyclic AMP, which is an intermediate step in what we refer to as a second messenger. This tends to amplify the whole process, and it's going to initiate more of a cascade of events involving a litany of different proteins within this cell that'll ultimately end up in some sort of cellular response. Oftentimes it's production of another protein, production of hormones, neurotransmitters, or any number of downstream processes. This is the cascade of events. Now, another option when that enzyme is activated is to turn on another second messenger. Calcium tends to be another common second messenger within these cascade of events related to G protein linked receptors. Calcium in this case, and I'm eliminating the whole host of steps or cascade of events that is happening, but calcium sometimes will actually bind to a channel, open up the channel, and allow ions, in this case sodium, into the channel, and that's going to depolarize the cell. To be very clear, that is very similar to what happens here. This is more direct. That is to say, the signaling molecule is binding directly to the receptor channel, and this, the opening of this ion channel, is requiring a whole cascade of events that is initiated by the G protein. Examples of G protein linked receptors are the cholinergic receptors known as muscarinic receptors. So once again, cholinergic receptors bind acetylcholine. Acetylcholine is a neurotransmitter that travels across the synaptic cleft and binds to a receptor. It's either going to bind to a nicotinic receptor or a muscarinic receptor. Muscarinic receptors are G protein linked receptors specific to acetylcholine, and nicotinic receptors are cholinergic receptors specific to acetylcholine. And then if we talk about norepinephrine, we have talked about norepinephrine. We said when norepinephrine is released from neurons, it binds to receptors on the myocardium or the heart muscle. It increases heart rate and force of ventricular contraction. Ventricles are a chamber within the heart that pumps blood throughout the whole body, at least the left ventricle. Norepinephrine binds to adrenergic receptors, and there are two types of adrenergic receptors. There's actually more than two types, but two broad categories: alpha receptors and beta receptors. That symbol right there represents alpha, and that symbol right there represents beta. I want to be clear, you never really want to write a B, because B represents something different than beta. So alpha and beta are specific types of adrenergic receptors, and they are both G protein linked receptors. Norepinephrine and epinephrine, because epinephrine, while it's a hormone, can by and large bind to the same receptors as norepinephrine, so they are very, very similar. They have very similar responses or outcomes. All receptors for norepinephrine are G protein linked. These are adrenergic receptors, alpha and beta. So when we start talking about hormones, we will learn that hormones travel through the blood. And the fact that hormones, which are signaling molecules very similar to neurotransmitters, the fact that hormones are traveling through the blood, one worry might be is that they're activating all cells in the body. But they are not, because cells have to have specific receptors for certain signaling molecules. So that is the workaround from preventing hormones from impacting all cells in the body when we only really want them activating certain cells. We have talked about specificity. That is to say, receptors in all of these cases need to be very specific to or let me rephrase it, receptors are very specific to what ligand or signaling molecule they permit to bind to them. Now, to be clear, there are things known as agonists and antagonists. An antagonist is some sort of molecule, could be endogenous from outside the body, it could be a pharmaceutical, or it could be pathogenic. That is to say, from a bacteria or virus, something foreign to the body intended to do harm. An antagonist will bind to receptors. They are capable of binding to receptors, say, that acetylcholine binds to. Antagonists will bind to receptors and prevent the downstream activity, which can be very detrimental because we need the downstream activity. Agonists will bind to receptors and cause the very same downstream event that the normal signaling molecule would achieve. A lot of bacterial infections like, for example, cholera and botulism interfere with G protein linked receptors, and that's how they cause disease or sickness, by actually binding to the receptors and preventing the downstream activity. I want to talk about some related factors or conditions related to receptors. One is saturation. Saturation is when all the available receptors on a cell are occupied. So we see these blue receptors up here. We see the ligand bound to the receptors. This cell is described as being saturated. There's no more receptors for any other signaling molecules to bind to. And I want to also mention that here we have, I believe, 5 receptors. There can be thousands or even 100,000 receptors on one cell, receptors specific to one signaling molecule. Sometimes there's a process known as upregulation that occurs, and this occurs when cells are, dare I say, desperate for increased metabolic activity. They need more receptor binding, so they place more receptors in the membrane to accommodate more signaling molecules. This happens in development, but it also happens during exercise. So we talked about insulin and insulin-dependent cells. Muscles are insulin-dependent cells that during times of no activity or no exercise, let's say, they require insulin for glucose uptake to move those glucose channels into the plasma membrane from the secretory vesicles. What exercise does 2 things. One, it upregulates the insulin receptors, so they're more receptive to the insulin. And 2, it actually upregulates the glucose channels, embedding them within the membrane. So those are 2 really good examples of upregulation. Now, downregulation is a thing as well. That is to say, if there's too much of the signaling molecule, the cells actually decide, that's too much activity, we're going to downregulate or get rid of receptors because we don't need that much metabolic activity. There's just too many of the signaling molecule out here. And type 2 diabetes is a great example of this. That is to say, a type 2 diabetic has excessively high glucose levels, generally due to lack of exercise and a poor diet, so they have elevated glucose levels. Because they have elevated glucose levels, they also have elevated insulin levels. And the downstream cells, these are the insulin-dependent cells, actually get rid of some of their insulin receptors, so insulin is no longer able to bind to all the receptors it wants to. Now, to be clear, the cells for type 2 diabetics may also become desensitized, which is very similar to downregulation, but it's really the receptors no longer recognizing the signaling molecule, or at least no longer responding to that signaling molecule. Diabetes also is related to downregulation when it's related to the brain. If you recall, glucose is the source of fuel the brain uses to make ATP. Now, the brain has a very important apparatus known as the blood-brain barrier. The blood-brain barrier is a very selective membrane around the capillaries that are providing blood or perfusing blood to the brain. And because our brain is so precious and important, it's regulating what gets in there, because we don't want pathogens, toxins, what have you, gaining access to the brain. So there's this blood-brain barrier that's very, very selective on what gains access to the brain. And certainly, the brain wants glucose. So there are glucose channels within the blood-brain barrier providing access to the brain. But in diabetics who have elevated glucose levels, there's so much glucose, the brain, specifically the blood-brain barrier, tends to downregulate or remove some of those glucose channels, because these diabetics oftentimes have elevated blood glucose levels. As a result, some of those glucose channels are taken away from the blood-brain barrier, because not all of them are needed because there's so much glucose in the blood. But then when diabetics, when they don't have sufficient glucose in their blood, or there's excessive insulin being released, now there's a low blood glucose levels. And this is why diabetics need to very closely monitor not only their glucose levels, but their insulin levels. So if there's excessive insulin, the insulin-dependent cells downstream are going to utilize the glucose because that insulin is providing the opportunity for glucose uptake into muscle cells, liver cells, and adipocytes. Or between meals where a diabetic has not eaten for a while, they have low blood glucose levels. And if their blood-brain barrier had previously downregulated receptors, they don't have as many glucose receptors as normal, and now this individual has diminished blood glucose levels, it's a bad combination. That is to say, not enough glucose transporters and not even enough glucose in the blood. So then there's a problem with mentation and level of consciousness with these individuals who need to get glucose into their system ASAP. So these are just some examples of receptors. Receptors are important, and we'll talk more about them as we move forward.